Scanning electron microscope SEM statement. Scanning electron microscope SEM is a technique used to produce high resolution images of the surface morphology of materials. It uses a focused beam of high energy electrons to scan the surface of a sample, producing a three-dimensional images. How does SEM work? It contains electron gun, column, sample chamber, detector, image formation. Electron gun. A beam of electrons is generated by a electron gun. Column. The electron beam is focused and accelerated through a column. Sample chamber. The focused electron beam is directed at the sample in the sample chamber. Detector. The electrons that interact with the sample produce signals that are detected by a detector. Image formation. The detected signals are used to form an image of the sample surface. Types of SEMs. 1. Conventional SEM uses a tungsten filament electron gun. 2. Field emission SEM FESEM uses a field emission electron gun, providing higher resolution and brighter images. Environmental SEM ESEM allows for the imaging of sample in a controlled environment such as high pressure or temperature. Cryogenic SEM always allows for the imaging of samples at very low temperatures. Applications of SEM. Material science. It is used in material science, biology, forensic science, quality control. Material science. Studying the surface morphology and composition of materials. Imaging cells, tissues and microorganisms. Biology. Forensic science. Analyzing evidence such as hair and fibers. Quality control. Inspecting the surface quality of manufactured components. Advantages of SEM. High resolution, large depth of field, non-destructive method. High resolution provides high resolution images of the sample surface. Large depth of field allows for the imaging of samples with a large depth of field. Non-destructive does not damage the sample during imaging. Limitations of SEM. Sample presentation, vacuum requirements, interpretation of images. 1. Sample preparation. Requires careful sample preparation including coating and mounting. Vacuum requirements. Requires a vacuum environment which can be challenging for certain samples. Interpretation of images. Requires expertise in interpretation the images and understanding the sample morphology. Types of signals in SEM. 1. Secondary electrons. Backscattered electrons. X-ray. Secondary electrons SE produced by the interaction between the electron beam and the sample surface. Number 2. Backscattered electrons BSE produced by the interaction between the electron beam and the sample surface and scattered back to the detector. X-rays produced by the interaction between the electron beam and the sample surface and to use it for elemental analysis.